Swayam Prabha. Digital India. Educated India.
Dear learners, I am Dr. Renuka Mainde, Corporate Nutritionist, Academic Counselor in IGNOU for their MSc DFSM course. Learners, the objective of present module are: discuss post-mortem examination of meat, explain rigor mortis, enumerate factors affecting rigor mortis, explain the process and factors affecting tenderization of meat, discuss different methods of tenderization of meat, discuss aging of meat and types of meat. Let's begin. Postmortem changes begins to appear once the animal is slaughtered. Postmortem examination and inspection refers to inspection of carcass and organs by veterinary doctor after slaughtering and dressing. It should be carried out as soon as possible after dressing. The carcass is examined for the evidence of bruises, injuries or discoloration and diseased conditions. The veterinary doctor examines carcass and organs to ensure that carcass and organs are fit for human consumption. During inspection, care should be taken not to contaminate the carcass and organs from deceased animals. The knives and other instruments used for cutting and examining organs, glands and tissues should be properly sterilized before and after use. A particular sequence should be followed during postmortem examination so that each carcass and the organs are checked thoroughly. Postmortem principles. Visual perception. First, the carcass and visceral organs should be examined visually for any visible abnormalities. Examination is done for any change of color, atrophy, hypertrophy, neoplastic conditions, etc. Palpation. The organs are palpated for any change in consistency, sliminess or gelation, cyst, etc. Incisions. The organs are incised if needed. This is done to examine any parasite inside organ and structural deformity. Laboratory test. These are done for confirmation and support the observation made by microscopic examination. While examining the organs of carcass, lymph node of adjoining areas must be examined. Let us understand what is rigor mortis. In conversion of muscle to meat, rigor mortis is the most important event. The muscle become inextensible or contracted and joints become stiffened after death, and this condition is called as rigor mortis. The stiffening results from the permanent cross bridges between actin and myosin filaments in the muscles. The cross bridges are the result of accumulation of lactic acid in the muscle, decline in muscle pH and coagulation of muscle protein. Characters of rigor mortis. The phenomena of rigor mortis is characterized by hardening and contraction of all voluntary muscles, loss in transparency of the surface of the muscles, stiffening of joints and slight rise in temperature by 1 to 1.5 degrees centigrade or more. After death or slaughter of an animal, blood supply of the muscle ceases, leading to severe deficiency of oxygen, energy, and vital elements essential for cellular activity, and an anaerobic environment in the muscle is created. During normal glycolysis, in the presence of oxygen, through a series of biochemical changes, the muscle glycogen is converted into adenosine triphosphate, carbon dioxide, and water. However, absence of oxygen favors anaerobic glycolysis, and due to action of lactic dehydrogenase glycogen is converted into lactic acid, thereby causing drastic reduction in ATP production. The lactic acid formed during the glycolysis goes on accumulating and lowering the pH of the muscles. The pH begins to decline from about 7 till 5 to 5.5. With the reduction in pH, ATP content of the muscle starts declining from its pre-Riger concentration of 5.9 micromoles per gram at pH 7 to 7.2 to near zero at pH 5.3 to 5.5. The level of ATP can be maintained for some time by resynthesis from ADP and creatinine phosphate, but the quality of ATP generated is very low and the overall level falls, leading to further reduction in pH due to lactic acid formation. This lowering of pH leads to weakening in the permeability of sarcoplasmic reticulum, which results into influx of calcium ions in the cells. Process of rigor mortis, calcium ion gets bound to troponin and causes conformational changes in the tropomyosin, resulting into release of the magnesium activated ATPase enzyme. Due to action of this ATPase enzyme, ATP at actin combining site on myosin and ATP at myosin combining site on actin are hydrolyzed. As a result, myosin and actin interact together to form cross-linkage as actomyosin complex, which cannot be disassociated. This actomyosin complex results in inextensibility and toughening of the muscles, making them rigid and non-contractile. This rigidity and toughening to the muscles is termed as rigor mortis. The loss of extensibility due to actomyosin formation proceeds slowly at first and then rapidly, after which it remains at low level. The onset of rigor mortis is accompanied by lowering of water holding capacity of muscles. Let us understand the pattern of rigor mortis. Acid rigor. The onset pattern of rigor mortis can be classified as acid rigor, which is characterized by long delay period and a short fast phase, and in struggling animals by a drastic curtailment of the delay period. Alkaline rigor, which is characterized by rapid onset of stiffening and by marked shortening of muscles. I'll just repeat this again. Intermediate rigor. This is characterized by the curtailment of the delay phase but not of the rapid phase in starved animals. There is some degree of shortening also. Rigor mortis starts from muscles of head and neck and then extends to rest of the body and finally the legs. In healthy animal, it starts after 9 to 12 hours of slaughter, stays for 20 to 24 hours and then passes. After 20 to 24 hours, the rigidity of the muscles cannot be maintained due to lowering of ATP concentration, since it is also required to maintain the stromal integrity of the proteins. Let us learn about tenderization of meat. Meat tenderness is one of the quality parameters that affects the tough muscles and changes into soft meat, which is characterized by increased palatability, juiciness, flavor of meat. This process is known as meat tenderization. Tenderness of meat is influenced by the breed, age, condition of nutrition and amount of muscular exercise of the animal. I'll just repeat this again. Tenderization of meat. Tenderness of meat is influenced by the breed, age, condition of nutrition and amount of muscular exercise of the animal. It mainly depends on amount of connective tissues present between muscle fibers and to lesser extent on the thickness of muscle fibers themselves. There are several methods by which tough meat obtained from different animals, especially aged domestic animals, can be converted into reasonably tender meat. Conditioning of meat. Conditioning is the natural process of tenderization when meat is stored or aged post-rigor. When meat is stored above freezing point at temperatures between 0 degree centigrade to 3 degree centigrade, all the changes that usually occur at higher temperature takes place but at a reduced rate. In the absence of microbial spoilage at low temperature, the proteolytic enzymes of muscle fibers bring about a desirable change, that is conditioning or the ripening, which is manifested by marked increase in flavor, juiciness and tenderness of the meat. The action of enzymes is almost completely inhibited when the meat is stored at temperature below freezing. Bacterial action does not bring conditioning. Tender cut process. In this process, tension on individual muscle is suspended carcass is reduced by selectively severing the bones and ligaments. The carcasses are suspended normally from Achilles tendons and thus it is easy to implement on commercial slaughter line. When the process is carried out within 45 minutes of slaughter, a significant improvement in tenderness has been noticed both in cattle and pig carcass meat. Meat tenderization methods. Electric stimulation. Electrical stimulation of carcass following slaughter is the new commercial method of meat tenderization. Electric stimulation is the application of sufficient current into the carcass during the slaughter process. Pulses of electricity are passed through the carcass immediately after slaughter and the current causes the muscles to contract and use up glycogen, ATP and creatine phosphate. It is done before rigor or in pre-rigor carcass. The electrical stimulation of pre-rigor muscle causes the carcass to undergo a rapid series of muscle contraction and relaxation. The rapid series of contraction and relaxation of muscle accelerates the rigor process. Advantages of electrical stimulation: improves the quality of meat from aged or spent animals, increases tenderness and reduces aging times, improves appearance and color of lean meat, can advance the process of rigor mortis, avoids cold shortening and thaw rigor. Mechanical tenderization. Mechanical methods such as needle or blade tenderization, stick maceration, tumbling, massaging, grinding, cubbing, etc. are effective methods for improving tenderness. Needle or blade tenderization is accomplished by passing the stick or boneless subprimals through the blades and or a bank of needles. Mechanical tenderization. The blades or bank of needles pass through the meat, severing connective tissues and muscle fibers, making the meat more palatable. A variable speed conveyor is used to advance the meat, while the up and down motion of the needle bank penetrates the muscle tissues. The best results are obtained when boneless subprimals such as boneless rib eye and boneless top sirloin are used. Mechanical tenderization. Grinding is very popular means of increasing tenderness of meat, especially beef. Ground meat is more powerful than steaks and roast due to its uniform texture and tenderness. Another means of mechanical tenderization is cubbing, where small blades of a cubber simply sever connective tissues in boneless retail cuts to break the tissue into smaller pieces. Tenderization by enzymes. Enzymes which have been used for tenderizing meat include papain, a proteolytic enzyme from papaya fruit, phycine, a protease derived from figs, bromelain, a proteolytic enzyme of pineapple. These tenderizers can be used in liquid or powder form, and in both cases at commercial or in the home. Tenderization by enzymes. Bacterial and fungal proteolytic enzymes act only on the proteins of muscle fiber, causing digestion of sarcolemma, disappearance of nuclei, degradation of muscle fiber, and eventually a loss of proteolytic enzymes of plant origin. Act preferentially against connective tissue proteins. Main feature of these enzymes are that they do not attack native collagen as it is denatured by heat during cooking. They break down the heat denatured connective tissues proteins to soluble hydroxyproline containing molecules. Methods of application of proteolytic enzymes. Dipping of meat in enzyme solution. Introduction of enzyme solution into meat pieces through fork holes before cooking. Pumping the major blood vessels of meat cuts post-mortem with the enzyme containing solution. Rehydrating freeze-dried streaks in a solution containing proteolytic enzymes. Pre-slaughter injection, that is given 10 to 15 minutes before slaughter, of solution 0.5 mg per live weight, 5 to 10% solution containing enzymes. High pressure tenderization. The application of pressure up to 150 megapascal to muscles and related systems have revealed responses of considerable magnitude and potential usefulness. The post-mortem glycolysis can be controlled to a greater extent, and it can be so accelerated that it is virtually completed within few minutes. Meat of any desired pH value is the usual post-mortem range can be produced. The pressure treatment also causes changes in other structure of muscle and results in tenderization. Z-line degradation is found in pressure-treated muscle fibers. This is due to the calcium ion dependent protease that is released when the mitochondria and the sarcoplasmic reticulum are affected by high pressure. A procedure has also been developed for pressure tenderization of post-Riger muscles. Tenderization by ginger extract. Ginger is widely used in household and restaurants for the preparation of various culinary dishes because of its spicy flavor imparting properties. Recently, ginger rhizome was investigated as new source of proteolytic enzyme of plant origin. Tenderization by ginger extract. Proteolytic activity of this enzyme was found to be many times greater on collagen than actomycin, thereby considered to be effective for tenderizing meat from old animals. Ginger extract also possesses antioxidant and antimicrobial properties, making it more beneficial for fast food facilities serving pre-cooked meat. Tenderization by marination of meat. The tenderness of meat and add taste variety to the meat components of meals by process of marination. The basic ingredients of a marinade include salt, acid like vinegar, lemon or soy sauce, and enzymes like papain, bromelain and ginger extract. Addition of little amount of oil seals the surface of meat from air and result in fresher and brighter appearance of meat for a longer period of time. Marination tenderizes the meat through the softening of collagen by the salt and hydrolysis and the breakage of the crosslinks of the connective tissues by the acids and alcohol. Meat is cut into small pieces or stripes to help complete penetration of the marinade into the center of the meat. Then, meat is mixed with marinating ingredients and kept in refrigerator for minimum of 4 to 8 hours for complete marination. Tenderization by cooking of meat. Cooking plays two different roles in tenderness of meat. The contractile protein myofibrillar proteins of meat become less tender due to coagulation during cooking process. On the other hand, major connective tissue protein, that is collagen, is converted into gelatin by cooking and becomes more tender. Cut such as steaks and chops from ribs and loin are low in connective tissues. Dry heat cooking like pan frying, broiling, roasting or barbecuing is recommended for such cuts because dry heat raises the temperature very quickly to develop the flavor of meat before the contractile proteins have the opportunity to become significantly less tender. Moist heat cooking such as braising is recommended for cuts like foreshank, round and chuck, which are having high amount of connective tissues, because application of moist heat for a long time at low temperature converts collagen into gelatin and makes the meat tender. Aging of meat. In the absence of microbial spoilage, the holding of unprocessed meat above freezing point is known as aging. It is frequently referred as conditioning, tenderizing, ripening or maturing of meat. By this process, muscle can be made soft. During the process of holding at 0 to 3 degrees centigrade, that is above freezing point, several changes occurs in meat at suitable rate. Aging is required for marked increase in flavor, juiciness and tenderness of meat. Aging is important for beef and buffalo meat, while pork and lamb do not require aging because they are slaughtered in the age while they are young and inheriting tenderness. Two types of post-mortem aging process are practiced commercially: dry aging, wet aging. Dry aging is the traditional process of placing an entire carcass or wholesale cuts without covering or packing in a refrigerated room for 21 to 28 days. The temperature required is at 32 to 34 degree Fahrenheit and 100 to 85% relative humidity with an air velocity of 0.5 to 2.5 meter per second. Too much humidity will allow excessive microbial growth, whereas too little will cause excessive shrinkage. Too high an air velocity, on the other hand, will result in excessive surface drying with resulting weight and trim losses. The main disadvantage of dry aging is the cost associated with these weight and trim losses. Wet aging. Wet aging is the aging of meat in vacuum bags, usually the middle meat, under refrigerated condition of 32 to 34 degree Fahrenheit. Obviously, humidity and air velocity are not necessarily requirements for proper wet aging. Because most beef is vacuum packed at the site of carcass fabrication, wet aging is the predominant method of post-mortem aging today. Significant changes in meat during aging. In the conversion of muscle to meat, natural enzymes found in the muscles break down specific protein in muscle fibers. Significant changes in meat during aging. This breaking of these protein strands, called myofibrils, by natural enzymes result in improved tenderness, protein denaturation, proteolysis, flavor enhancement. Flavor enhancement during aging. During post-mortem aging, ATP is broken down to mononucleotides, that is AMP, and IMP, inosine monophosphate, which produce inosinic acid and hypoxanthine, enhancing the flavor of meat. Besides, there are production of some flavor compounds such as hydrogen sulfide, ammonia acetaldehyde acetone and diacetyl by microbes, most particularly by yeast, and in long-term aging by molds. Let's conclude, learners. Post-mortem changes begins to appear once animal is slaughtered. Post-mortem examination and inspection refers to inspection of carcass and organs by veterinary doctor after slaughtering and dressing. In conversion of muscle to meat, rigor mortis is the most important event. The muscle become inextensible or contracted and joints become stiffened after death, and this condition is called as rigor mortis. After death, slaughter of an animal, blood supply of the muscle ceases, leading to severe deficiency of oxygen, energy and vital elements essential for cellular activity, and an anaerobic environment in the muscle is created. The onset pattern of rigor mortis can be classified as acid rigor, which is characterized by long delay period and short fast phase, and in struggling animals by drastic curtailment of the delay period. Alkaline rigor, which is characterized by rapid onset of stiffening and marked by shortening of the muscles. Intermediate rigor, this is characterized by curtailment of the delay phase but not of the rapid phase in starved animals. There is some degree of shortening also. Meat tenderness is one of the quality parameters that affect tough muscles and changes into soft muscle, which is characterized by increased palatability, juiciness and flavor of the meat. The process is known as meat tenderization. Tenderization of meat can be done by various methods like mechanical, electrical, applying enzymes, to name few. In the absence of microbial spoilage, the holding of unprocessed meat above freezing point is known as aging. Thank you, dear learners.